Hello everyone. Today's topic is that biomanufacturing, bioprinting, and organ on chip. Biotechnology is undergoing a transformative era with the rise of advanced platforms such as biomanufacturing, bioprinting, and organ on chip technologies. These breakthroughs combine biology with engineering to create innovative solutions for healthcare, pharmaceuticals, and sustainability. From harnessing living cells for large-scale production to fabricating tissues through 3D bioprinting and simulating human organs on microchips, these technologies promise to reshape research, diagnostics, and treatment strategies. By bridging these gaps, traditional biology models and real-world applications, they open new possibilities for personalized medicine, regenerative therapies, and efficient drug discovery. So, continuing with that, my teammate will continue on biomanufacturing.
Introducing to biomanufacturing, the key concept that utilizing living cells to produce valuable bioproduct, it enhances the efficiency and sustainability. It also introduction to advanced technology like robotics and AI to transform traditional manufacturing production, and growing focus on sustainable practice in biomanufacturing uh, and biomanufacturing materials and methods. Exploring a key component technology used like biological system and raw material to create product. Technical include fermentation, cell culture, and enzyme technology. This method are enable the production of complex biomolecules, enhance efficiency and sustainability in various industries. Uh, the application of biomanufacturing. Uh, there are three types: ba- pharmaceuticals, tissue engineering, and food production. In pharmaceuticals, production of complex and drugs and biological efficiency. In tissue engineering, they used a field allowed to create a biological sustain to restore, maintain, and improve the tissue function. In food production, it focus on food safety and sustainable by improving the nutritional quality and reducing waste. The advantage of biomanufacturing. Uh, the three main focus are sustainability, cost efficiency, and customization. In sustainability, it is eco-friendly practices to reduce environmental impact. In cost efficiency, the cost while maintaining high quality outputs. In customization, the tailored solution to meet specific needs of various markets. The disadvantages of biomanufacturing are scalability challenge, regulatory hurdles, and cost cost implication. Scaling up a production process can be difficult and inefficient. Cost impact, high cost for material process can be limit fund growth. So, over to Rajshekhar.
Hello everyone. Today I'm going to present on bioprinting, an emerging technology that is shaping the future of medicine. In introduction, it is a specialized form of 3D printing that uses bioinks which contain living cells, biomaterials, and growth factors to create biological structures. Unlike normal 3D printing with plastics or metals, bioprinting aims to replicate tissues and in the future even complete organs. This is especially important because the shortage of organ donors is the major global problem. Materials and methods. The process begins with bioink, which is usually made from natural substance like collagen, gelatin, or alginate, or either from a synthetic material such as PEG, polyethylene glycol, PLA, and so on. Bioink provides a supportive environment for cells to survive and grow. There are different methods are used for bioprinting. Inkjet bioprinting spray, droplet of cell suspension, similar to how normal printer works. Coming to the application point of view, regenerative medicine. Printing of cells, bone, cartilage, patches for heart or liver repair, drug testing and disease model. Highly detailed organs model can aid in surgical planning and education, and also in cancer research. Tumor models help to study cancer progression, tested drug therapies. Advantages. The major advantage of bioprinting are it allows patient-specific solution, that is precision replacement of cells and materials, complexity, allows for the creation of integrate tissue structures, and also potential to solve organ shortages. Coming to the disadvantages, at the same time, there are some disadvantages associated with the bioprinting. There is a lack of suitable bioinks, material limitation that fully mimic natural tissue properties. Usually, the high cost setup is required for the operational purposes, and also there's a lack of facility and ethical issues and safety are a very concerned about it. Over to my teammate Rohit Kumar.
What if I say you can see a pumping heart on a chip? Yes, you heard it right. This technology is called organ on chip. Well, this is Rohit. Join me with learning organ on chip technology. Well, it is a microfluidic device that mimics the structural and functional units of human organs. It combines microelectromechanical systems with tissue engineering, where it provides 3D microenvironment for cells under which dynamic fluid flow is established. The main aim for creating this organ chip is to recreate physiological responses and predict drug efficiency and toxicity better than traditional 2D cultures and animal models which are currently available. Moving on to methods and methodologies. The commonly used material is polydimethylsiloxane. It is widely used because of its attractive properties such as commonly available. It is highly biocompatible with biological tissues and transparent. Being transparent is one of the biggest advantages because it can be directly exposed under microscope and it can be viewed. Materials like thermoplastics and hydrogels are also used, but not as widely used as PDMS. The fabrication methods generally involve soft lithography, microfluidics for control flow, and also it is basically integrated with sensors like electrical, optical, and mechanical systems. The commonly used cell types are primary human cells, induced pluripotent stem cells, which have the capability to proliferate into any cell lineage, and organoids. The common method that is used is the cells are seeded on the glass plate, and soon perfusion flow is allowed to flow on the glass, whereas where the tissue gets integrated with the surface, therefore forming a tissue interface. Example is alveolar capillary in lung chip. Moving on to the applications, specific biomedical research involves disease modeling, where cancer on chip is developed, as well as COVID-19 lung model is developed during COVID-19 for doing research. And the toxicology studies involve drug-induced liver injury and nephrotoxicity studies. Pharmaceutical industry is also uses this technology to screen the drugs as well as to establish safety testing for the clinically proven drugs. And personalized medicines also use this for patient-derived cells on chips. And this is mainly done to replace or to reduce the need for animal models. Specific organ examples include lung on chip, liver on chip, heart on chip, gut on chip, and multi-organ chips. Moving on to advantages and disadvantages, it is closer mimic of human physiology than the currently available 2D cultures and animal models. And real-time monitoring of cellular responses can be done, which is at a cost-effective price and high throughput compared to animal studies, which takes comparatively longer time, as well as it enables personalized testing with patient-derived cells. The disadvantages includes still it lacks full organ complexity. Complex cascade of enzymatic reactions cannot be studied, as well as fabrication requires expertise as well as infrastructure. And limited long-term culture stability is also one of the major drawbacks associated with it. Hence, the scalability is also a great question in organ-on-chip technology. Well, handing over the session to Sanjay.
So my teammates gave a good explanation about the three topics involved here. So I would like to conclude the three topics So biomanufacturing is driving sustainable and cost-effective production of medicines, food, and biomaterials. So bioprinting is opening new frontiers in tissue repair, regenerative medicine, and personalized drug testing. And organ chip provides accurate human-like models, reducing animal testing and improving disease research. Together, these technologies are not just innovations, but a foundation for the future of healthcare and biotechnology, making treatments more precise, research more reliable, and solutions more sustainable, and avoiding human errors. Thank you, everyone.